Question: chemical shift. ANS chemical shift is a fundamental concept in nuclear magnetic resonance spectroscopy. It refers to the variation in the resonant frequency of atomic nuclei when they are placed in a magnetic field and subjected to radiofrequency radiation. The chemical shift is expressed in ppm and is used to identify and characterize the chemical environment of specific nuclei in a molecule. Chemical shift values are influenced by the local electronic environment around the nucleus being studied. Different types of atoms and their positions within a molecule result in unique chemical shift value, allowing researchers to determine the chemical structure and composition of a sample. In proton NMR, for example, the chemical shift of a hydrogen nucleus, proton, is influenced by its proximity to other atoms and the electron density in its vicinity, providing valuable information about the molecule synthesis structure and bonding. Factors: 1. Electronic environment. The local electron density and the chemical structure surrounding the nucleus can significantly affect its chemical shift. Electronegative atoms or functional groups nearby can result in deshielding, shift to higher ppm values, while electron-donating groups can cause shielding, shift to lower ppm value. 2. Magnetic field strength. The strength of external magnetic field used in the NMR experiment affects the chemical shift. Higher field strength lead to better resolution and larger chemical shift dispersion. 3. Isotopic mass. The chemical shift of a particular nucleus may vary slightly depending on its isotope. For example, hydrogen, 1H, and deuterium, 2H, nuclei will have different chemical shifts. 4. Solvent. The solvent in which the NMR experiment is conducted can influence the chemical shift. Some solvents have characteristic peaks in their NMR spectra which can affect the observed chemical shifts of the sample. 5. Temperature. Temperature can impact chemical shift. NMR experiments are often conducted at a controlled temperature to minimize variation. 6. Instrument concentration. Accurate calibration of the NMR instrument is crucial to obtaining precise chemical shift values. Improper calibration can introduce error. 7. Sample concentration. The concentration of the sample can affect the signal-to-noise ratio and in turn, the accuracy of chemical shift measurements. Importances: 1. Structural determination. 2. Compound identification. Chemical shift patterns are like molecular fingerprints that help chemicals identify specific functional groups and the arrangement of atoms in a molecule. 3. Purity analysis. Impurities or the presence of multiple compounds in a sample can be detected by analyzing the chemical shift patterns. 4. Kinetics and dynamics. Chemical shift can provide insight into the dynamics and conformational changes of molecules over time. By monitoring changes in chemical shifts, researchers can study reaction mechanism and rates. 5. Quantitative analysis. 6. Protein structure. 7. Quality control. 8. Drug discovery. NMR is instrumental in drug discovery by characterizing the binding of small molecules to biological targets, helping design and optimize pharmaceuticals. 8. Environment and food chemistry.